so as I said before, I'm going to take up some experiments that I did while in Seattle for a year, some experiments and simulations, and this work is in collaboration with Professor Wayne Xu at the Fred Hutchinson Cancer Research Center. The problem we were interested in is the evolution of cooperation. So cooperation has consistently evolved on many biological scales. Where's the link?
Oh, I'm sorry. Sorry.
Okay, so cooperation has consistently evolved on many biological scales. So genes cooperate to form genomes, organelles cooperate to form cells, and cells cooperate to form large organisms. But what we're interested in is how does this complex collective behavior fit into the picture of survival of the fittest, where each individual is fighting for their own survival, and collective behavior is seemingly not preferred. So a few things have been expected to either be detrimental or helpful to cooperation. One of the big things that's been covered in the literature a lot is defectors. So cooperative systems should be very susceptible to defectors. That's because cooperators usually pay a cost for cooperation, so their fitness is slightly less. So if you take but do not give, then you have a slight advantage, and these defectors should be able to overrun the entire population, thus destroy cooperation. However, having a defined spatial structure to interact in can allow for isolated cooperative groups to exist. So, for example, in many game theories, and in particular the spatial prisoner's dilemma, it was shown that if you have a bunch of cooperators and defectors and they're all mixed up, then the defectors will always overrun the cooperators. However, if you have them on a spatial grid and you're only playing games with the neighbors around you, then isolated cooperative groups can exist stably. So we can find many examples in nature. Microorganisms can form clusters of cooperators in biofilms, colonies, etc. So a biofilm is, for example, the plaque on your teeth. If you have any, that's a bacterial biofilm, where the bacteria on the inside of the colony are kind of protected from corrosive chemicals or anything like that. So Professor Shou created a system that she deemed Cosmo, which is an anagram for cooperation that is synthetic and mutually obligatory. So it's an engineered system of budding yeast, so Saccharomyces cerevisiae. So this is just beer yeast, bread yeast, except it's lab strain, of course. So we engineered them so that they were deficient for one of two nutrients, either lysine or adenine. So those genes have been knocked out and they can't produce their own. And they can't survive by themselves. These adenine or lysine must be supplied externally if these cultures are going to survive. We've also labeled them with red fluorescent protein and green fluorescent protein so that we can tell them apart under the microscope. So this deficiency has been complemented, though, by overproduction of the opposing nutrients. So if you're deficient for lysine, then you overproduce adenine. Basically, that feedback loop has been knocked out, so you just keep producing and producing and producing and never stop. Now, most of that overproduced nutrient is actually stored inside what's called a vacuole, which is kind of like the stomach, and it's usually released upon cell death. I just want you to remember that. So usually when they die, they kind of burst open like a piñata, and all the goodies come out. So you think these two yeast strains can't survive by themselves, but if you put them together in a co-culture, one might think that they should cooperate in order to survive. So there's enough nutrient floating around that both strains should be able to grow.
But so they're stored in the vacuoles, but before they die, are they accessible then to the...
Maybe a little bit. It's not clear. I mean, it's not clear if they release exactly on cell death or just prior to cell death, but either way, usually you don't see... Well, I'll show you the data, actually.
Okay.
It's released close to, if not upon cell death. Okay, so here's some characteristics of the Cosmos system that I want to show you first. So here we have the red fluorescent strain and the green fluorescent strain, and these are their populations, function of time. This was measured by flow cytometry, facts. What I want to tell you first is that in the pink area there, the cells experience an initial growth phase and transfer to a minimal medium. So we have to grow these cells up overnight, and they have some nutrients stored inside of them. So whenever you start the experiment, they always double a few times. So that's what you're going to see in many graphs, and that's what that is. The lysine deficient cells, they die much faster than the adenine deficient cells. And we believe that's because lysine is a amino acid. So without that, there's a lot of proteins that you can make that really affect your cell functionality. But adenine is a nucleotide, so really it just affects their ability to reproduce, but they can still go on functioning pretty well. Eventually it catches up to them when they start dying. And initially there's always this kind of delay in the death phase for the adenine deficient guys. So they don't really start dying until about 70 hours after the experiment has started. So I just want you to remember that point as well. So this is how they behave when they're by themselves in cultures alone. But when we mix it together above a certain density, we get the population as a whole to grow. So these lysine deficient guys undergo a wave of death, and then they start growing because these guys here have started dying, and it just kind of wiggles back and forth on and on until the culture saturates. And saturation is defined by they've run out of sugar. So they've kind of reached their maximum capacity in the culture. So, but cold cultured system will grow to saturation. Okay? So if we want to come up with a dynamic model, we're not trying to fit this data point by point at all. We're just trying to get an idea of what's going on. We might start with just the growth equations for each strain. So first order differential equation. But what we need is a good model for what the net growth rate is. So they gotta grow, and they gotta die, and those growth rates have to depend on the concentration of nutrients. One of the simplest models you can have is this kind of Monod model, or it's kind of similar to Michaelis-Menten equation. So you have a growth term which depends on the concentration of lysine L for these guys, and then a death term. So a concentration equals 0, then they're just dying, and at a very high concentration the growth rate saturates. So what does that look like? So that looks something like that. So below a certain concentration, they're dying, and above a certain concentration, the net growth rate is positive, so they're growing, and that saturates out. And then this constant Ka or Ka is kind of just their affinity for that nutrient. So there's no really good physical explanation for this model, except that it's very simple, but actually it works pretty well, because we can measure in this part of the model here. I actually measured the adenine deficient strains. I measured fed cells and starving cells, so we starve them, just let them hang out a few days without any sugar, just to see if there was a big difference between if they were fed or starving. But in either case, we get an affinity of about 0.1 microgram. And this was done using time-lapse microscopy over a few days' time. So there's two other equations that we need to know if we want to try to model this, and that's the differential equation for the adenine concentration and the lysine concentration. So those have two terms in them. There's a consumption term, so they're consuming nutrients, and then there's also a production term. So the death rate there, you can see in the production terms. That's why I was telling you that they kind of release these nutrients upon cell death, so that's why it actually is there. So they're consuming nutrients and producing nutrients upon death. And BA, PA, and BL, and PL are the molecules. They're just a conversion term. Molecules consumed to produce a new cell, and molecules produced upon cell death. Okay?
What is L?
L is lysine concentration, and A is adenine concentration.
What is rho then?
Rho is the density of lysine deficient. I guess I should have colored it red to make it more clear.
That would work.
Rho is the density of lysine deficient cooperators, and rho A is the density of adenine deficient cooperators. So that really is what defines each strain, because they're auxotrophic for these nutrients that can never change, so they just will die without them. So that's how they
So there's how many inhibitory terms there, right?
there's no inhibitory terms. Here, their growth just depends on the individual concentration, and without that, they'll die. Their consumption also follows the same kind of chemical kinetic form with this conversion term, and then the production kind of depends on the death rate of the opposite strain. So DA refers to these guys, and DL refers to these guys. So the production of adenine depends on how fast they are dying, and the production of lysine depends on how fast these guys are dying. I know it's a little confusing. Is that clear? Let me go back to this slide just quickly. So the lysine deficient strain here, when they die, they are releasing adenine. And these adenine deficient strain here, when they die, they're releasing lysine, so that the other strain can grow. It's kind of a circulatory system.
Well, it's interesting. If you look at your plots, it sort of looks like stepwise increments
Yeah, and those oscillations tend to damp out after a while.
Yeah.
We don't think it's evolution going on. I'm not sure exactly. I mean, there is evolution that happens pretty quickly, and that's a whole other bag.
But it's almost as if you take that image from the left and just sort of keep ramping it up, sort of
yes, yeah.
an order of magnitude or so.
That's right.
Yeah.
Yeah, so they definitely require waves of depth to continue growing, which is why they kind of depend on some of that. But the oscillations do damp out, actually.
Interesting.
Okay? So here's our model. It's 4 first-order differential equations in time. It's nonlinear. There are a couple of things we can do with it. Let me just show you some of the solutions, what they look like. If you start at a high enough cell density, right, about 10 to the 4 cells per milliliter, then they go through waves of death, but then eventually the system starts growing at some growth rate that's definitely less than the maximum of yeast. So you don't see oscillations here. So like I said, we're not trying to fit everything point by point. This is just kind of a general model to kind of describe the consumption and production of nutrients.
The other seems to have some very short, the growth happens in a very short interval.
Yeah. You mean right there?
Not about in the experiment. I mean,
the initial growth rates that I was talking about?
The jump, they seem to happen in a short...
That's right. Well, I did originally in the model, in the model I originally had some heaviside functions in there so that they would only release upon cell death. But it turns out that it simplified everything a little bit. It really doesn't change the results if I remove those.
No, I don't mean putting things by hand. Heaviside or step function.
You're just saying, observing in the data, it looks like the steps are pretty sharp there. That's what you mean?
Yeah.
Yeah, they are. They're growing close to maximum in these range here. So the doubling time of yeast is about an hour and a half, and you can look on the graph here, and you can see that this experiment's
over 200 hours.
Yeah, almost 2 weeks long. So when they're growing at maximum, it looks like almost like a vertical line in this graph.
But there are 2 time scales: the death rate, if you like.
Yes.
And then there is much quicker.
That's right. So they use up their nutrients immediately. They grow. Many cells grow very quickly, exponentially, and then they die slowly, and then grow exponentially very quickly again. But like I said, these oscillations tend to damp out, and it's more of just like a straight line if you keep following that graph and keep carrying the culture forward over and over and over again. You just keep putting them into a new tube every other day or so. Okay, so the point I want to make is it's
Sorry, about 5
yeah,
minutes.
go ahead.
5 minutes, sorry.
Oh, really? I only have 5 minutes left?
Yeah, about 5 minutes, yeah.
Okay. So above a certain density, they will grow. And if you start them at too low of a density, however, they will not grow. So there's bifurcation in the behavior of the system in the sense that if they're not started at a high enough density, not enough nutrient is released, and they never just make it to a net positive growth rate. So this can be captured in this phase diagram here. We can solve the equation 10,000 times or so, and we can get gray points are the ones where the culture was inviable, and purple points are the cultures that were viable. So I'm going to reiterate again, this is a well-mixed liquid culture because I'm going to talk about spatial structure in just a minute. So we can extract the viability boundary. So below this boundary, if you start with these initial conditions, the culture will be inviable, and above here, it will be viable. There's one other thing that defines this phase boundary, and that is the fact that cells are discrete quantities. So you have to put these vertical lines and vertical and horizontal lines on the boundaries, basically saying that the density times the volume has got to be greater than 1 cell, right? You can't have less than 1 cell. And here it's greater than 9 cells, and that's kind of a subtle point. It's actually due to that delay into death I was talking about. They have to die first, and then when the other guys start dying, then they can grow. So.
Yeah, before this one, the pattern looked pretty symmetrical.
Yeah.
And this shows an asymmetry between the 2.
That's right. But we also have data on this. So all the purple points were cultures that were always viable. All the replicas that we did were viable. All the light purple points are the ones that were sometimes viable, and then all the gray ones are replicas that were just never viable. Mm-hmm. So it seems that the model is doing pretty well in describing the system behavior right now. So we think we understand the well-mixed situation pretty well and kind of the dynamics of what's going on. If we want to think about spatial structure, the simplest thing you can think about is growing them on a petri dish, right? So this is kind of just, instead of having the cells all mixed uniformly, let's confine it to one surface. Well, I'm going to commit, usually when you think about spatial structure, you think about spatial structure in the X and Y dimension, right, kind of on the surface. But what I'm going to convince you here is what really ends up mattering is kind of the depth of the agar. If you evenly distribute the cells on the surface, let me just show you what a movie of time-lapse growth, where we image the cells on a surface of agar gel from below, because the agar is mostly transparent. And then we focus on the agar surface. We have to do a bunch of autofocus. In a week's time, we can get green fluorescent and red fluorescent images. And so you can see all the cells distributed on the surface here. Each cell is about 5 microns in diameter, 3 to 5 microns. That's a movie for you. So this is in hours here. So the green guys, the adenine deficient guys grow up, and all the red guys are dying. You can see them disappearing, but eventually, after about 70 to 90 hours, somewhere around there, the red guys just explode. And only a few guys are the progenitors in the next generation. So these big cooperating pods form, and I kind of stopped the experiment there at 140 hours because the light was so bright it was saturating the camera. But if you look at one of those pods, after about 300 hours, we get some confocal imaging on those, and they're pretty amazing.
Wow.
This is a combination of a Z-stack of a bunch of images, and then you combine them together so you kind of get this 3D effect. So this is looking from above now. So we're really not sure what's going on with these pods right now. What I'm going to tell you about in just 1 second and briefly, since I don't have much time yet, is that the viability really only depends on the depth of the agar. So this is one of our future directions, is trying to understand how these are cooperating in this column, in this intracellular matrix. Okay? So if we think about one-dimensional spatial structure just for a minute, we got cells uniformly mixed and distributed on the surface on a gel with a height h. So the nutrients must diffuse rather than be mixed homogenously. And a dimensional analysis will reveal a length scale when the growth is limited by diffusion. So chi is the diffusivity of the molecule, and R average is that kind of average growth rate that I was showing you in those plots earlier. After the system is viable and it's reached steady state growth rate, that's what R average is. So if the average cell spacing is much less than this, then the cells in any given area see mostly uniform concentration in the XY plane, and then only the Z dimension is important. That's kind of evidenced by the fact that in that movie I showed you all the red guys grew up simultaneously, so they're all seeing the same concentration at the same time. For our system, this length scale is about 1 centimeter, so we should see difference in behavior when H is kind of getting past 1 centimeter. So here we have the spatial model. I'm not going to go into this. We have cells per centimeter squared now, so I'm using sigma instead of rho. Here are 2 growth equations, the same as before. Here we have the diffusion equation for lysine and adenine. I guess those should be partials. Sorry about that. And consumption now is a flux on the surface. Consumption and production is a flux on the surface. And at the bottom of the gel, we have 0 flux. So I just want to show you we can make phase diagrams before with viable and inviable regimes. Here we're plotting the height h on the x-axis of the agar gel and the total number of cells per square centimeter, sigma L plus sigma A on the y-axis. And I'm constricting my cells to 1-to-1 ratio just to make things simple. Of course, you can change that parameter as well. You can start out with very skewed ratios and see how that changes. We can extract the boundary here. If the diffusivity was infinite, this would be just like a well-mixed culture. You would get this straight line. So here, right about 1 centimeter, it starts to roll over, and the cells just don't care how it is. So they don't care if the agar is 10 meters below them or 10 centimeters or whatever. So I'm taking data on this. This is after 500 hours. The purple points, or the blue points are ones that were viable, and the magenta ones were inviable. This experiment needs to be repeated, I should say. This is kind of preliminary data, but it seems that the model is working pretty well right now. If we change parameters, say reduce PL by a factor of 3 or reduce the death rate by a factor of 2, we can shift this line around, but it really doesn't change that length scale of where it rolls over at. We can also think about separation of cooperators. We can take cells on top of an agar gel. We can cut it in half with a razor. We can place the cooperators on the opposite side and then ask, well, what would this look like? They shouldn't be able to cooperate anymore if they get far enough apart. In fact, that is the case. We can produce a phase diagram for this as well with the partners on opposite faces. So as the gel gets very deep, they get farther and farther apart. They can't communicate anymore. So they're just never going to be viable anywhere past this line right here. We can put these two together. So this is the one with partners on opposite faces, this line here. This is the one with the partners on the same face. And then they kind of diverge right at that length scale that I was showing you earlier, which is a result of the balance of growth and diffusion. Okay, so sorry I had to go so fast towards the end there, but I'm a little bit time limited. So quickly, summary and my outlook. The dynamic model of the Cosmos system reproduces the well-mixed phase diagram and agrees with the data variables, so we're happy about that. And the spatial model accurately predicts the viability boundary on the 1D agar gel. And 1D, I just mean that only the height really ends up mattering. So for spatial dimensions much less than the diffusivity divided by the average growth rate to the 1/2, the viability is essentially the same as liquid culture. That's one of the messages I want you to take home, is that there's this important length scale, and below that, nothing's really, spatial structure doesn't really matter that much. Viable conditions, on agar, the viable conditions develop these cooperating pods of those cell types, and this is one thing we want to look at. This is the instigators of the spatial structure in the XY dimension, which is a much more difficult problem. But that's something that we're starting to look at. And one of the future directions, I just want to come back to my introduction real quick, and we really want to add defectors to this culture to see, okay, so how do these defector cells, ones that just don't overproduce anything, right? They need nutrients, but they don't produce any. How do they affect it? And the research was supported by the NIH and ICAM, of course, and the Tech Foundation. So thank you very much for your time.
Yeah, thank you. Steve?
Did you ever think about laying these out in stripes?
Yes, these are many things that there were so many experiments to do. First, we just wanted to do the simplest and spread them evenly and say, can we understand this? So I should say that a lot of this is just trying to quantify our method so that we can quantify evolution. So what we're really interested in is, okay, we made this cooperative system. How's it going to, it's fake, right? We've engineered it, but how's it going to evolve now? What kind of changes? Is it going to get better or worse? So now we know, basically if we can measure the parameters in the problem, we can test them by putting the yeast in liquid culture or in a spatial environment, and then we can see how, and then we'll know if it changes happen in the future, quantify what kind of changes must have happened to facilitate that. So we want to do stripes, checkerboard, all these different things. We just haven't got around to it yet. And those pods make everything much more difficult because it's apparent, at least I think it's apparent to me, that the diffusion of the nutrients inside a colony cannot be the same as it is in a free agar gel.
Right.
Because it's a lot of polymers in there, the viscosity is much higher, it's just going to be a much more difficult problem. So.
Other questions? Yeah, Jim.
Should I think of this as an interesting model system for a nonlinear dynamics problem, or should I think of it as trying to get at some biological system where you might be wondering about how cooperativity works or doesn't work?
Well, certainly the bigger picture is we're trying to understand how a system that might evolve from inception, how a cooperative system might evolve from inception. I came at it with a nonlinear point of view because of my background. Because I really saw the phase diagram and the oscillations, and I was really interested in all that. And it's just kind of come together into one picture now. So we can use the modeling and the numerics and the quantitation to kind of understand how things are evolving later on. I would think about it maybe both ways.
I mean, is there a system in nature that you would point to and say, this might help us understand a problem that's been puzzling us about cooperativity and so on and so forth?
Well, there's certainly examples of cooperation in microorganisms. I think one example that was, I think, in a paper by Jeff Gore was about the sucrose metabolism in yeast and how they, I think it was some of the sugar that they end up, some of the sucrose that they end up degrading into glucose escapes. So they have to be able to degrade it, but a lot of that escapes, so they're paying a cost in order to cooperate. I'm not too familiar with it right off the top of my head, but this is kind of a fake system that we engineered to really trying to get at the heart of cooperation, whereas that is actually a real thing that yeast do, break down heavier sugars and more simple ones.
Just a comment. I know Bob Austin has been playing around with sort of bacterial colonies that can evolve much more quickly
Yes.
with cooperation and defectors, sort of, you know, I forget what he called it, but sort of, you know, good guys and bad guys. And the point is that you can really see evolvability in these colonies. But those are much simpler organisms than yeast. So being able to do the same thing with yeast could be really quite interesting. Steve?
Yeah, just one other question. In the conditions where you start to see the pods, doesn't that change the viscosity, the diffusion coefficient?
Oh, I think I just had a comment about that. That's why I haven't really delved into attempting to model those, because
Let's change it a lot.
Right.
it's going to be a nightmare, yeah. So I'm not sure what to do about that. But my model basically predicts the viability when the cells are more or less still in a monolayer on the surface. Beyond that, I really can't say anything.
Sorry, Randall? No? Okay. Well, I have a suggestion. Since we have 2 more of these talks by our young scientists, and we want to be able to give them full attention, why don't we take about a 5-minute break where we can stand up, perhaps use the restroom, breathe, and then let's come back in for the last 2 talks. And thank Justin.